Well, thanks very much, Dan and Terry, for the kind invitation to be here today. You already heard that my wife largely ignores everything that I do, so with that in mind, I should probably have relabeled this why AFib is really cool and why should you care. Um, you've got 15 minutes to learn everything you ever wanted to know about atrial fibrillation. It's most common arrhythmia. It's really bad. Leads to a risk of heart failure, stroke, death, dementia. We have terrible treatments for getting you back into a regular rhythm, but great treatments for protecting against stroke. And it's getting more common. About 1 in 3 of you, if you're over 50, are going to have atrial fibrillation the rest of your life. So you may want to keep that in mind as we go through some of these slides. And this is not just in the red states. This is an increasing problem throughout. I mean, this is the increasing prevalence of atrial fibrillation. This is true of other cardiovascular diseases in these same regions. Just to drive home the point about, these are the 2 best drugs we have to treat atrial fibrillation in a randomized controlled trial. This is amiodarone, 75% of the prescriptions of drugs to get you in a regular rhythm. It's about 40 to 50% effective at 3 years. Second best drug, 20%. Ablation procedures do better. We'll save that for another day. What are the available genetic? So I hang out with the Broad in addition to being at Mass General. So we've used genetics to try to pick apart some of the underlying mechanisms. We have a lot of different genetic resources. It's always terrible to follow study talks on breast cancer, schizophrenia. Every time I think we're doing well with some of our genetic resources, they look pretty puny compared to these other studies, but about 120,000 AF cases for GWAS from a lot of different sources. Increasing genome sequencing support from which we receive both from the TopMed and the co-funding from the CCDG programs, and then ongoing discussions to do larger scale exome sequencing in the CCDG project. Just a brief vignette on where we stand for each of these efforts. This is Carolina, who led our latest meta-analysis, 65,000 individuals, about 100 loci from which we can construct these scores. This came out just last year. Similarly, this is Seung Wong, who looked at our initial genome sequence data. Really simple experiment, just cases, controls. Were there any genes that emerged with a loss of function variant out of the noise? There was one gene, Titin, well known in dilated cardiomyopathy and many other forms of inherited cardiovascular heart disease. We saw about a doubling in the cases of early onset AFib compared to controls. What was interesting is that the younger and younger that you were, so here's the controls, about 1%, cases about 2%. The younger that you are, the greater the increasing frequency of finding a Titin loss of function mutation. About 1 in 20, 5 to 7% of people under 40 with AFib have a Titin loss of function variant. So that's your crash course on the genetics of AFib. I think my mandate today, we've learned, we've seen a number of different, like many GWAS studies, we've seen a number of different genes, the challenges linking it to mechanism, of course. But I think the mandate today was how can we think about using polygenic risk to inform clinical outcomes or research questions for AFib. One is, can we just use polygenic risk to predict AFib itself, either in the global population or individual subsets of the disease? Can you use a polygenic risk to as essentially a biomarker for the disease? And if so, why would you? And then what about differential outcomes, these horrible things I talked about, heart failure, dementia, death. And then lastly, as Dan mentioned, and this is one of the advantages of going later in the day, is you can change your slides on the fly. You can look at the interaction between common and rare variation. So I'll try and touch on those quickly. The first to realize for AFib is that it's, I think we all see these Manhattan plots that are forests. Dan, you named this a Dubai plot. This is what it looks like if I have the unadjusted. Was it Carlos? Yes. There's one dominant locus for AFib that sticks out and has a dramatic increased risk of disease. And that's something we've known about for a while. We've also, this was going back to 2010 when we just took 3 SNPs at this top locus, put them together in some pretty primitive haplotypes. But what we saw was about 1% of the population that had a massively increased risk of the disease, something that has been refined in the years since. And then we can do that in a more elegant fashion. This is Lu Chen Wang and Sarah Price. They took one of the early GWASs for AF, and I'm going to almost skip all of the methods today, other than just to get to the punchlines. But they derived 30 different candidate scores. They fit those scores to the UK Biobank to find the optimal score, and then they reapplied that to Framingham to see what was the impact of the genetic risk on the subsequent development of atrial fibrillation. And again, summarizing their work in 2 images. The first is that if you're down here, if you're 55 years of age and you're of European descent, because Framingham is almost exclusively of European descent, this is the risk of developing atrial fibrillation the rest of your life. It's at 37%. That's increased over the last 15 to 20 years. Probably both with a sad reflection on the state of the health of the average American with increasing cardiovascular risk factors, also increasing recognition of the disorder itself. So then one, lifetime risk is high. The second is that it's strongly modified by polygenic risk. Now we just took the score that fit best in the UK Biobank, applied it to Framingham, and then just divided it crudely into 3 different groups, high, medium, and low. So if you're in this middle group, obviously the middle group stayed the same at 37, but if you have a high polygenic risk, you go to nearly a 1 in 2 chance of developing atrial fibrillation. If you're on the lower end, you're down to a 1 in 4. So you have this 20% stratification, basically just with high, medium, or low risk of disease. Why would we care about that? Well, there are some individuals, if you're at the low end, where you may have, and we see these commonly, they walk in, they have no risk factors for AFib, and they're going to be at the high end of the polygenic risk. And you have other individuals that have modifiable features that are within this. Just because you have a high polygenic risk doesn't mean that there aren't modifiable risk factors within this. So the other area that I would think of, so we can use AFib to help, we can use polygenic risk to try to identify individuals at high risk of developing AFib in the population, but can we also think about using it as a biomarker in select populations? And the one example that I think is probably the most compelling is in the realm of cryptogenic stroke. Why cryptogenic stroke? So strokes from AFib, obviously AFib, you can develop a blood clot, travels to the brain, that's a stroke, that's a cardioembolic stroke, that's really bad. That's down here. Strokes from AFib, they're more disabling. You're more likely to die from them, and yet we have a good treatment. So if we can recognize the AFib in advance, get you on a blood thinner, we can prevent the strokes. Unfortunately, about a third of strokes coming into the hospital are of indeterminate or cryptogenic. And yet we know that there's AFib within these. If we put monitors into people, about a third of people in a 3-year time frame will develop AFib. But what you want to figure out is who within this group has AFib and who doesn't, because those monitors are expensive. Each one's $5,000+ plus the monitoring costs. And so if you can figure out which group has AFib, why not, you can then more effectively treat them, and you can prevent unnecessary treatments as well. So, and it's also important because you could imagine just, well, if you have a cryptogenic stroke, why not just give everybody a blood thinner? That's been done. The trials are universally negative. And so could we try to stratify the population? And if you were at that top 1% of a genetic risk for AFib and you had a cryptogenic stroke, well, you'd certainly want to have more invasive monitoring. So that's been done. Nice work by Steve Lubitz, Sarah Pewlett, and Chris Anderson. They've gone to the SIGN consortium where they've very carefully evaluated subtypes of stroke, the same ones that I showed you on that plot a moment ago. And they've looked at this a few different ways, but one, they took they looked at the correlation with the AF genetic risk scores. And then just a threshold, they've compared the AFib and the SIGN consortium to the polygenic risk of AF from an earlier study. And what you saw was a strong correlation between the two, no correlation just with educational attainment as a background control. You saw the same relation between AF variants and cardioembolic stroke. You'd expect that, but not with other forms of stroke. And then a weaker signal, but still present for this cryptogenic stroke population. So regardless of the specific methods, and Ahmed will talk more about this, you can take, this is, that was pruning and thresholding, this is LD-pred, but you can imagine just if you have a bell-shaped curve for your disease for AFib, why not just take off this this tip of the, this tip of the highest risk, and then think about further screening methods. Well, the other cool thing about AFib is that we have really neat ways to screen. So one is you can just put a patch on someone. Little patch, just a sticker on the chest, records every heartbeat for 2 weeks, and that will, with a very high resolution, pick up AFib. Downside is it's only 2 weeks, so it's not a very high yield if you've had a stroke. There are a lot of these devices. I'm by no means an expert on all screening modalities, but they're common in our world in electrophysiology. This is a little device you can just touch your fingers to. It'll send an EKG, that information to your phone. It will give you a little green thumbs up, or it doesn't give you a thumbs down. It just gives you a red and says, maybe you ought to go talk to your doctor. But it's very, very good at picking up AFib. This is an Apple Watch. I'm sure you've seen plenty of press reports about that. We'll come to that in a second. And then this is an injectable little monitor. You come into Mass General right now, you have a cryptogenic stroke, and we don't find any other obvious cause. We just inject a monitor into you. And that little monitor will record every heartbeat for up to 3 years. We pick up AFib, we're going to start you on an anticoagulant if you've had a stroke. So at that point, we can alter treatment based on the detection of the disease. This is already in routine clinical care. So is this, and a lot of your patients have this. Drive that point home. This is this Apple Watch is basically getting a signal from the back of your wrist, and then you're closing the loop to form an electrogram, a single lead electrocardiogram, by just touching it to the little dongle or dial on the side. This is what it looks like in the app. We'll find plenty of AFib, and it's been an amazing uptake with this particular device. Apple and the Apple Heart Study, I don't know if you've kept up with it, but the UK Biobank, I think everybody in the room is familiar with, 500,000 people. Apple Heart Study, they just sent a little message out to people that had previously bought Apple Watches and said, do you want to be in a study to detect stroke? 419,000 people participated. So same order of magnitude as the UK Biobank, and it went to someone's wrist. They found a lot of strokes. They also found a lot of noise. If you went to the ACC, meaning it's very good for picking up a normal rhythm. It's actually pretty good for picking up AFib. It doesn't do well with the stuff in between. As someone who still sees patients with AFib, Dan and I were talking about this over a drink last night. This is the part that scares the hell out of me, is that, just send all of these to your doctor. They can find my email. I don't want 600 pages of Apple reports. But it comes at a cost, any of these screening technologies. I'm not an ad for Apple. There are plenty of these others that are out there. Fitbit has the same technology, so does Google Fit. This is a Cardia device. The specific devices don't matter. This screening's already happening. Your patients are walking around with it. Some of you probably have it on your wrist at the moment. We're already doing large-scale screening. Steve Lubett's character, if you walk into Mass General right now and you're over 65 years of age, we've randomized half of our clinics to say, do you want to be screened for a stroke? We'll screen them for AF on that moment. That information is done by, that test is done by the medical assistants. It goes direct into Epic. It's available for the doctor to review. Reflexively, they can order a monitor or do an EKG. So that ability to embed this type of screening technology into the medical records already in current practice. And this is just, we're randomizing the two groups. So I won't touch really on the, differential outcomes. Sorry. Maybe I'll just skip that. We're going to touch really on the differential outcomes because that's still early work. I think taking polygenic risk to look at, say, how you respond to a medicine, an ablation, whether you have any increased propensity to bleeding, better protection against a stroke, that's still areas of active investigation. And we're just starting to look at polygenic risk and then adding it to this rare variation. Mainly that's just a limitation of sample size at the moment. As we have much larger genome and exome sequencing data sets, we'll be well suited to do that. So hopefully I've convinced you that AFib is really cool. The main thing, we've identified a lot of different genetic variants associated with the disease. We think that polygenic risk can be used to identify high-risk individuals. We hope that it'll help us refine cryptogenic stroke. obviously, as Adrian said this morning, there are already risk prediction models for atrial fibrillation on the Framingham website. This should not be considered in isolation. Just like any other cardiovascular disease, just knowing someone's age, gender, and then and hypertension status, you're going to capture the bulk of the information for any one of these diseases. And then just to be aware, if you aren't, that screening of AFib is increasingly widespread, and you'll see more and more of it in the years to come. These are all the folks that work with me. I've had great collaborations with many of the folks, including Amit, Sek, and others at the Broad. Thanks very much. Happy to address any questions.
Okay, we have a couple minutes for questions.
Patrick, that was a great talk. In terms of the Titin mutations, do you see left ventricular dysfunction, or are they in individuals with stone cold normal function?
Yeah, it's a great question. You get both. So that what you see is a spectrum. We, in that particular study, had intentionally stratified folks that were young, healthy, no overt structural abnormalities. But the closer you look, the more that you'll find. I think that the Titin variation will be kind of a spectrum going from overt myopathy to an arrhythmia like AFib, and then some people in the middle where you have, and that's already been studied from some of the literature in the cardiomyopathy literature, where folks have looked at Titin loss of function carriers in Framingham and in other populations, and then looked at with cardiac MR imaging and found dilated left ventricles, subtler forms of LV dysfunction, and I'd expect that in some cases AFib will just be the canary in the coal mine. But a lot of the people that we saw had longitudinal follow-up of many, many years, and they did not go on to develop AFib. Some did not go on to develop a myopathy. So you had some that just had AFib, some that went on to develop a myopathy, others that didn't.
Yeah, could you speak to this last point, I think, which is a very interesting one in heart disease, where there's pleiotropy and clearly overlap. One disease, so to speak, begets another or is an increased risk. How do you look at that in trying to disentangle that, or do you not worry about that? You know, in cancer, it's very rare. It's a tiny fraction of people that get more than one cancer, and usually when they do, it's a consequence of chemotherapy and/or radiation, you know, or a very rare germline mutation. But you know, your last comment was really raising a really fascinating question of sort of the clarity of what's the risk for AF versus this, or does it really matter to the patients? Wouldn't you rather be able to say, well, you're at risk for these 5 things in this differential order, and cumulatively you have a 38% increased risk due to all of these factors?
Yeah, I think some of it goes back to ultimately when you get back to the patient level. When I'm seeing someone in the office, it's really how is it going to modify their care? You know, I think that in the stroke populations, you pick up AFib and they're going to be on a blood thinner. So there's a discrete test, or there's a discrete change in clinical practice that you'll go to. You do bring up an important point, though, of shared contributions. You know, especially as you see larger and larger genetic analyses, you know, are you really doing a GWAS just for AFib, or also hypertension, diabetes, other forms of cardiovascular disease? You have to be mindful of that. And a lot of those commonalities have been well worked out from some of the prior epidemiologic work in the area. I mean, you don't need you don't need a polygenic risk to tell me that hypertension's the leading population attributable risk for developing AFib or many other forms of heart disease. But it's a very important point.
David?
Along the Titin
Yep.
path, we find many Titin variants, and I'm wondering if those to which you are attributing atrial fibrillation have an earlier age at onset, or are they different than what might be called garden variety atrial fibrillation? Definitely, and in this population, that was where we saw the increasing risk with an earlier and earlier age of onset. So in the overall our overall population started at 65 or under. So mindful that most AFib is actually 72 to 75 years of age on average. So we've already picked off a small group to focus on. Obviously, we focus there because the younger onset, the higher the likelihood of a genetic contribution. But it's about 5 to 7% of folks under 40 years of age. But it would be minute if you just looked at 70-year-olds. It'd be a pretty small fraction of the disease, like minuscule.
Thank you. We've got a discussion period after the last talk, which is going to be on genomic risk through the lifespan from Amit Kara.